Welcome to the lecture on biological molecules. So we're going to look at the four main functions of biological molecules. We won't really talk about functional groups much, but you can look those up in the textbook or in the PowerPoint lecture as well. We're going to look at carbohydrates, lipids, proteins, and nucleic acids. So here I have a nice big diagram here for you. So first thing we're going to do is talk about carbohydrates. What in the world are carbohydrates? Now in order to lecture with this setup, I'm going to be bouncing back between screens here a little bit. So be patient with me moving from screen to screen. Alright, so up here I have drawn a pretty elementary drawing of carbohydrates. Carbohydrates are made up of carbon, as all these molecules will be, a carbon skeleton, and that's what this is right here, is a carbon skeleton. So we have a carbon, a carbon, a carbon, a carbon, and those lines in between represent the covalent bonds between the carbons. So our carbons are going to be made up of carbons. They're also going to be made up of hydrogens and oxygens. And you'll often see this right here, which is a type of functional group called hydroxyl. And that's very common to hydroxyl, and that should be a Y there. Sorry. Hydroxyl. And those are very common to carbohydrates as well as to alcohols. I'm going to clear the ink off there. Now, when we're talking about carbohydrates, we all know that pastas, breads, potatoes, those things consist of carbohydrates. So do your candy bars, pop, sugar that are found in those are carbohydrates. So let's talk about the different types of carbohydrates. So you have simple carbs and you have complex carbs. Simple carbs are going to be your basic sugars like your sucrose, your glucose, your fructose, your galactose, your maltose. Those are simple sugars. Complex sugars, complex carbs, are your starch, glycogen, cellulose, things that are found in your breads and your potatoes and your pasta. Those are complex carbs. They're large molecules is basically what that means. You have simple carbs, which are small molecules. Complex carbs are large molecules. Now, when we're looking at simple carbs, we have monosaccharides, which is what that is right there. Mono meaning one. Okay, so mono meaning one, and saccharide meaning sugar. So one sugar. Now, a lot of times what we want to do is we take two monosaccharides. So here we have glucose and fructose, both monosaccharides. We're going to put them together. Now, how do we put two molecules together? Well, we go through what we call a H2O getting pulled out of here. We call it a dehydration synthesis. So what happens is H2O gets pulled out of the molecule. And if you look on page, I believe, 47 in your textbook, it actually has a good diagram of that. But you pull H2O out of your molecule, and you get a disaccharide. We put two monosaccharides together and we get a disaccharide, meaning di meaning two sugar. Okay, two sugar, and that should be a G. Two sugars equals a disaccharide. Now if we did this tons of times that had many sugars, we would get a polysaccharide, meaning many sugars. Okay, so that's how we organize carbs. We have simple sugars, such as monosaccharides and disaccharides. I apologize for the sneeze. And then we have polysaccharides, which is many sugars strung together, and that's what this represents. Each one of these little yellow right here is a sugar all strung together. This would be your complex carb. Now, carbs are used in the body for energy. We break them down to make ATP, which is what I've drawn down here for you. It's stored in the body as glycogen for animals. We store glycogen in the body. We break it down in our liver and our muscle cells to create ATP. ATP is just the energy molecule in our body, most common one. This is its pre-use state, where it has a lot of potential energy. And then when we use it, it becomes that right there, which is ADP. Because what happens is we rip a phosphate off, which we'll get into in chapter 6. But that's ATP. So carbohydrates are mainly used to create ATP. carbohydrates are also used to burn lipids. We have to have carbs to burn lipids. So a lot of times people go on no-carb diets thinking they'll burn their fat, but we have to have carbs to burn lipids. Alright, next let's talk about lipids. What are lipids? Well, I'm going to go to the blank page next door here and show you a little bit. There's two main things we're going to talk about. First is triglycerides. Glycerides is a type of a lipid, most commonly in our body. And we have, the reason it's called triglycerides is it has 3 fatty acids, 1, 2, 3, and a glycerol backbone. Glycerol, Glycerol. So that's this molecule, right? This whole thing right here. There's glycerol, and it's bound with 3 fatty acids. That makes a triglyceride. Now, when we look specifically at a fatty acids, there's 2 types. There's saturated, okay, and if I just have single bonds between all the carbons and these hydrogens coming off, and so hydrogens would come off at every point possible here. This would be a saturated fatty acid. Saturated fatty acid. Now, if I want to draw an unsaturated fatty acid, there would be a double bond. And when you have a double bond, like that one, you can't get as many hydrogens, so it's unsaturated. So this would be monounsaturated, meaning 1 double bond. If there was more than 1, that would be a polyunsaturated fatty acid. Now, unsaturated fatty acids, because of that double bond, they end up looking more like this rather than a saturated, so that's unsaturated. And a saturated would look straight like that. The saturateds are able to stack on top of each other. They'll be your butters, things that can stack. They're also not very healthy because they stack in our arteries. They stick to the artery walls. Unsaturated are going to be your liquid at room temperature, and they're going to be healthier for us because they don't stack. So make sure you know those 2 types of fatty acids. You can find those on the triglycerides. For example, we could have a triglyceride that has a saturated and then an unsaturated and then a saturated fatty acid. So you could have different types. So this would be a monounsaturated triglyceride. Okay, you could have polyunsaturated triglycerides. All right. We also have phospholipids, which is another main important type of lipid. Hormones, steroid hormones are types of lipids that are very important. So like cholesterol, testosterone, estrogen, those are type of lipids as well. If we go back to the previous page here, you can see this right here is are phospholipids. Phospholipids are very important. They look like this. 2 tails. These tails are fatty acids. And then the top right here is a phosphate molecule, and that's a type of functional group. Very important because it makes this molecule, having that phosphate and the fatty acids makes it polar in a way. So if I clear the ink here, the top parts of the phospholipids right here are hydrophilic, and the middle part in here is hydrophobic. And that's very important because that makes our cell membrane have unique properties that allows us to control the substances that come in and out. So that's very important for our body, having those unique characteristics from the phospholipids. All right, next we're going to look at proteins. So proteins are made up of amino acids. Now the neat thing about proteins, if we go to this next page, making up of amino acids are made up of 2 main functional groups. We have an amino group, and that's the central carbon of the amino acid, and then a carboxylic acid group. And what's the name of these? They're amino acids. Where do they get their name? I wonder. Probably from carboxylic acid and amino. Put them together, you get amino acid. Okay? These 2 right here, this one and this one, are functional groups you need to know. If I gave you an amino acid, you'd be able to tell me that those are the 2 types of functional groups in an amino acid. Now what happens is we put amino acids together to make proteins, and that's what I have up here. There's an amino acid, there's an amino acid, there's an amino acid, there's an amino acid. We string those together, again, through a dehydration synthesis. That's how we build these molecules. Alright, and we bond the two amino acids together. This bond is a special type of covalent bond we call a peptide bond. Okay? When you have tons of amino acids together, you have a polypeptide, which is what can become a protein. A polypeptide, meaning tons of amino acids, because you have tons of peptide bonds, so that's going to make tons of amino acids strung together, which will make a protein. Now let's look again here at this other page. So if we have a bunch of amino acids, and let's say these are amino acids, this is what we call the primary structure and the primary sequence. So primary structure of protein. It's the actual amino acids and their sequence. So how are they laid out? How is that initial structure of amino acids made? That's the primary sequence and that primary structure. And that's the most important structure of proteins because it determines its overall structure and function. This primary sequence does. The secondary structure is localized structure. So you can have little corkscrews along the way, or you could have pleated sheets and some more corkscrews. Okay, and those are secondary structures. And again, that's just localized structure along the way, along the polypeptide chain. And then you have tertiary. Tertiary is if you have once you have those localized structures, the whole polypeptide chain will ball up, fold up into a ball. And then the last one is quaternary. And quaternary is if you have a bunch of polypeptides together, more than one, okay? More than one polypeptide together, that's quaternary. Now you could have a protein just at the tertiary structure, but most proteins are going to have the quaternary structure. You can have both though. Okay, so those are the four structures of protein. The most important is the primary because that determines the rest and its function. Now protein has a lot of functions in the body. For example, structural, contractile, they're enzymes. Enzymes help speed up all the reactions in our body. Some hormones are proteins. It's structural part of our body with our muscles. They also have help our muscles contract. They're found in our membrane, which I've shown us down here. Okay, they can be receptor sites. They can be channels that allow certain things to go through. Okay, proteins can also bind with lipids. We have lipoproteins. These actually carry our cholesterol around. You've seen LDL and HDL. Low density up here, lipoprotein, high density lipoprotein. They carry our cholesterol through our body. We also have glycoproteins, which is sugar and protein together. Okay, so protein's a very important part of our body. It's found everywhere. It has tons of functions. Okay, I can't even list them all right here. They transport, they help things move through in and out of cells. They're structural. They're functional. So a lot of things are at the responsibility of proteins. Now real quick before I move on to nucleic acids, I want to make sure I touched on one thing that I might have missed earlier. Is when we do the dehydration synthesis to build larger molecules, we can also reverse it and put the H2O back in and break it back down into two molecules through what we call hydrolysis. So make sure you understand that as well. So dehydration synthesis, take water out, build, and then hydrolysis, hydro meaning water, bring water back in, lysis meaning break apart. So bring water back in to break something apart. Okay, so those are reversible reactions there. Alright, finally let's talk about nucleic acids. There's two types of nucleic acids we're going to talk about, DNA and RNA. DNA is a two-stranded molecule, looks like that. And along its backbone here, it has phosphate bound to a sugar with what we call a nitrogen base coming off. This right here, this part that I just drew, is found all along these strands of DNA. That's what makes up the DNA here. Okay, along each one of these strands. This right here is called a nucleotide. And that's terrible handwriting. Let me rewrite this here. Nucleotide. And a nucleotide is a phosphate bound to a sugar with a nitrogen base coming off, and we'll call that adenine. There's 4 types of bases for DNA. It's adenine, guanine, cytosine, and thymine. And remember, DNA is double-stranded. It's found inside of the nucleus of the cell, and looks like that, how it's all strung out like spaghetti. Okay? And DNA can't leave the nucleus, so we have to have something else to send the message, to send the blueprints from DNA to the cell, so everybody knows what they're doing. And that's where RNA comes into play. So along these strands here, RNA comes in and takes messages along the way. So if we go back here, so if we, let's say this section right here wanted a message, we would build an RNA molecule off of that. That molecule would then travel out of the nucleus to what we call a ribosome, which is right there. That ribosome would then read the message and build a protein. We'll get into the more detailed process of that in transcription and translation in chapter 14. Now, in order to build that RNA molecule, we actually have to split across apart the double helix. So what holds the double helix together? Why are we able to split that apart so easily? Well, if I get rid of these lines I just wrote here, where these bonds are here, where these bases are here, I mean, along the way here, so this is a base, there's a base, there's a base, there's another base on the other side, and they're bound together through hydrogen bonds. And that's what holds the 2 strands together, is hydrogen bonds between the bases. And the bases are paired together, so A goes with T and G goes with C. And that holds the 2 strands together in a double helix. So if I clear this ink, so if, let's say, here's our strand here, let's say we have an A over here, adenine, there'd be a thymine base over here, and they'd be bound together. If guanine was over here, cytosine would be over here, and they'd be bound together by hydrogen bonds. Okay, those are hydrogen bonds. And that holds the DNA double helix together. RNA doesn't have to have this because it's just single-stranded. RNA also doesn't have thymine. It has A, it has G, and it has C, but instead of thymine it has U. Those are for RNA. So DNA is found in the nucleus. It's a double helix. Okay, it's got your nucleotides. RNA also has nucleotides. RNA leaves the nucleus and is used for protein production. Right, it goes to the ribosome and then it codes the message for proteins. All right, so that's a nice overview of your biological molecules. You have carbs, you got your lipids, your proteins, and your nucleic acids. Make sure you also touch up on your functional groups as well. And I'll see you next lecture.